Welcome to Rootkeep, where the root leads back to a metal humanity has trusted for 6,000 years and a practice erased in a single decade. In 1924, Mayo Clinic surgeons reported a dramatic drop in post-operative infections using silver-based protocols. In 2011, University of Arkansas researchers confirmed it kills over 99% of E. coli in minutes. Between those two dates, hospitals abandoned the practice entirely. The question is why? At the Mayo Clinic, a group of surgeons once tracked infection rates after surgery with unusual rigor. Their records from the mid-1920s describe a trial of silver nitrate washes applied to wounds. The numbers are clear. Infection rates fell from nearly 1 in 5 to about 1 in 8. It wasn't a miracle, but for an era before antibiotics, it was a measurable advance that caught the attention of hospital committees across the country. Nearly a century later, a University of Arkansas lab revisited the question with modern tools. Their findings were striking. A 5-part per million solution of silver ions wiped out more than 99% of E. coli in under an hour. At higher concentrations, the bacteria vanished in minutes. The effect was not subtle, and the data left little room for doubt. This kind of result can seem like a modern breakthrough, but the roots stretch much deeper. Archaeologists working along the Levantine coast have uncovered Phoenician amphorae lined with silver, their inner surfaces still stained with traces of preserved brine and rosemary oil. These ships carried not just cargo, but a practical understanding. Water stored in silver vessels resisted spoilage on journeys that lasted months. Centuries later, Hippocrates, writing in the 5th century BCE, prescribed thin sheets of silver for cleaning wounds, pairing the metal with honey and wine. He wrote that the application drew out putrefaction and seemed to speed recovery, an observation echoed by Roman and Persian physicians who favored silver-coated instruments and servingware for the royal courts. The pattern is hard to ignore, from Levantine traders to Greek healers, from frontier-era Americans boiling silver dollars in drinking water to Russian Orthodox rituals that immersed silver crosses. The metal's reputation as a safeguard against decay and infection was more than superstition. It was tested, repeated, and quietly built into protocols that survived for generations. The evidence is scattered across centuries and continents, but the logic is consistent. Silver, in controlled doses, could tip the odds against infection long before the world knew the word antibiotic. On the American frontier, medical certainty was rare, and improvisation was routine. Diaries from Oregon Trail immigrants in the 1860s describe a simple ritual: boiling a silver dollar in a water pot before drinking, especially when illness swept through the camp. The logic was practical, not mystical. Families saw that water stored with silver stayed clearer, and the habit spread by word of mouth, recorded in letters now held by state archives. This was not just folklore. The same principle appeared in Russian Orthodox rites, where silver crosses were dipped in holy water, a tradition later shown to impart a measurable antibacterial effect. As medicine professionalized in the late 19th century, silver's reputation shifted from campfire wisdom to clinical trial. In 1881, Carl Creed, a German obstetrician, published data from over 5,000 births. A single drop of silver nitrate in each newborn's eyes reduced cases of blinding ophthalmia from nearly every untreated infant to just 0.2%. The numbers were so stark that maternity wards across Europe and North America adopted the protocol within a decade. Silver had moved from the margins to the center of hospital practice. By the early 1900s, pharmaceutical companies recognized the demand. In 1902, Bayer launched Argyrol, a colloidal silver solution marketed for wound care and mucous membrane infections. Competing products like Protargol and Colargol soon followed, each claiming superior purity or stability. These were not patent medicines in the modern sense. Silver itself could not be patented, but their formulations were standardized, and their instructions appeared in the United States Pharmacopoeia and Merck Manual. Hospital formularies listed dozens of silver preparations, with recommended concentrations and dosing intervals. Johns Hopkins surgical records from the 1930s document the use of silver-coated catheters, reporting a 50% drop in catheter-related sepsis compared to untreated devices. Clinical trials in the Lancet and Mayo Clinic proceedings tracked infection rates, adverse reactions, and long-term outcomes, building a statistical case for silver's inclusion in routine care. By 1938, silver was woven into the fabric of hospital medicine. Its use was neither fringe nor experimental. Yet this ascent set the stage for a reversal, one driven not by a sudden loss of efficacy, but by changes in regulation, economics, and the arrival of new patentable drugs. Silver was not banned outright. Reframed, delisted, forgotten, it faded from protocols as swiftly as it had entered them. In 1938, the landscape of American medicine changed with the passage of the Federal Food, Drug, and Cosmetic Act. For the first time, the law required that all drugs, new and old, prove their safety before reaching patients. The language was sweeping, but its effect on silver was subtle at first. Colloidal silver solutions, once a staple in hospital supply rooms, now faced a new kind of scrutiny. Manufacturers needed to submit formal safety data, and the FDA gained the authority to block products that could cause harm, especially those linked, even rarely, to argyria, the blue-gray skin discoloration that haunted silver's reputation. The timing coincided with a scientific milestone. In 1928, Alexander Fleming discovered penicillin. By the early 1940s, wartime urgency drove the US government to fund a massive scale-up of antibiotic production. Pfizer, Merck, Squibb, and Eli Lilly received federal contracts to deliver penicillin to military hospitals. These new drugs were not only effective, they were patentable. The purification processes, the fermentation strains, and the finished antibiotics themselves could be protected for decades. In contrast, silver compounds, long known, chemically simple, and already in public use, could not be patented. No company could claim exclusive rights or guarantee a return on investment. The economic logic was inescapable. Pharmaceutical firms shifted research budgets away from silver and toward antibiotics, where every dollar spent promised proprietary control and government-backed demand. Hospital committees, faced with new regulatory expectations, found it easier to adopt antibiotics with clear manufacturer-supplied data than to defend legacy silver protocols that now seemed outdated or risky. Formularies quietly dropped silver listings. By the end of World War II, penicillin and its successors had become the first-line standard, while silver, never banned but now lacking commercial or institutional champions, began to disappear from medical practice. The break was not just scientific, it was structural, driven by the interplay of law, profit, and policy. Silver's story became one of reframing and erasure, not outright rejection. In 1947, the United States Pharmacopoeia quietly removed colloidal silver and its compounds from official drug listings. No ban, no scandal, just a line gone from the manual that shaped hospital medicine. The American Medical Association soon echoed this shift. In a 1950s editorial, its editors described silver's use as a relic of pre-antibiotic medicine, urging doctors to adopt penicillin and other new drugs instead. The message was clear. Silver had been recast from a hospital staple to a medical afterthought, its clinical status reduced not by a collapse in evidence, but by the machinery of regulation and the economics of the patent system. Yet the biology did not change. Decades after its institutional erasure, researchers began to revisit silver's effects with tools unavailable to earlier generations. In 2013, James Collins and his colleagues at Boston University published a study that reignited debate. They found that silver nanoparticles, when combined with conventional antibiotics, could lower the dose needed to kill drug-resistant bacteria by as much as 1,000-fold. The synergy was not a marginal effect. E. coli and MRSA strains that shrugged off antibiotics alone were rapidly cleared when silver was added at low concentrations. Collins's team traced this effect to silver's ability to disrupt bacterial membranes, making them more vulnerable to drugs that would otherwise fail. The evidence kept building. In 2018, a study at the University of Southampton tested silver-impregnated surfaces in hospital intensive care units. Over 6 months, these surfaces showed a 2-log reduction in bacterial contamination compared to ordinary plastic and steel, a drop of more than 99%. Infection rates on wards fell by nearly a third. In 2020, a meta-analysis published in the Journal of Antimicrobial Chemotherapy pooled dozens of clinical trials and confirmed that silver dressings reduced wound infections by about 30%, with benefits seen even against antibiotic-resistant organisms. Meanwhile, NASA engineers verified that silver ion water purification could prevent bacterial growth in the microgravity of the International Space Station, confirming that the effect held even in extreme environments. These results did not erase the risks. The dangers of argyria, irreversible skin discoloration caused by unregulated high-dose silver ingestion, remained real, especially outside clinical oversight. But the scientific reappraisal was decisive. Silver's antimicrobial power had persisted, even as its place in medicine had been rewritten. The story was never one of scientific failure, but of institutional memory loss and shifting incentives. What was once reframed, delisted, and forgotten proved under scrutiny to have never truly lost its edge. Silver was not banned outright. It was just reframed, delisted, and forgotten. Today, it quietly persists in burn units and water systems, evidence never fully erased by regulation or commerce. As antibiotic resistance surges, the boundaries of medical orthodoxy shift again. The past may hold answers we're only beginning to reconsider.